Chapter 18 is kind of a continuation from where we have been. And then after this week, and actually a little bit of next week, I don't think we'll get through chapter 18 in 2 lectures, but we might. Then we'll do a rapid departure to the respiratory system and spend quite a bit of time there. So chapter 18 just finishes up the cardiovascular system. We've looked at blood, we've looked at the heart, and now we're going to look at blood vessels. So blood vessels are going to be the conduits. So the heart is the pump. The blood is what is being propelled. And the blood vessels are going to be the conduits or the delivery system. And it will begin and end at the heart. So that would be the circulatory system. By definition, arteries carry blood away from the heart. Arteries are usually highly oxygenated. The exception is going to be that pulmonary trunk and pulmonary arteries. So pulmonary arteries carry deoxygenated blood, and pulmonary veins will carry oxygenated blood. So the pulmonary circulation is reversed. But for the systemic arteries, they will all be highly oxygenated. Capillaries are going to be very thin and allow for exchange of nutrients and gases and waste with the tissue cells. And veins, by definition, carry blood to the heart. So in all veins, except for the pulmonary veins, they are going to be deoxygenated. The larger vessels are going to contain 3 tunics, and we'll talk about when we start to lose tunics along the way. But we'll start with the larger vessels, and the larger vessels are going to have all 3 layers. So the tunica intima is a simple squamous endothelium. It usually has a basement membrane, but not always. And it is continuous with the endocardium. So the innermost lining of the heart is going to continue through those blood vessels and be one continuous smooth surface for blood vessels to flow through. Tunica media is going to be comprised of smooth muscle, so circular smooth muscle, and elastin, so elastic fibers. This is the thickest tunic in arteries, and it can be responsible for vasoconstriction and vasodilation, but to differing degrees depending on what the vessel size is. The tunica externa is made of loosely woven collagen fibers, and we'll show this in the histology section. There'll be kind of these lighter pink collagen fibers. By the collagen is going to provide strength to this layer and allow it to anchor to surrounding structures. Collagen fibers can be densely woven, like in tendons. They can be kind of haphazard, or they can just kind of have a looser but still organized structure. And that's where we are with the tunica externa. This allows for lymph vessels and nerves to feed through them, that collagen mesh. For the larger blood vessels, the tunica externa is too far from the lumen to get diffusion of nutrients and waste. So it has something called the vasa vasorum, which is going to supply the blood vessels with an exchange location. So this is just showing the tunica intima is going to be this simple squamous epithelial tissue. It's continuous with the endocardium, so it's also called endothelium. Tunica media is smooth muscle and elastic fibers. Tunica externa is collagen fibers, and then this vasa vasorum on the exterior is going to feed back to the blood vessel to keep it nourished. We'll talk about this capillary network kind of after we've talked about arteries and veins. It will be that the interchange location for between the capillaries and the tissues, but then it also connects the arterial and the venous systems. So this should be looking familiar. You saw this in lab. The artery is going to have, this is the tunica intima. It's like a light pink, looks kind of like, it's kind of rigid, like has little ridges. That's kind of part of the histology aspect. It's this innermost light pink. And then the darker purple is that tunica media. And then these light pink fibers are going to be collagen fibers of the tunica externa, along with elastic fibers. So it's hard to see the tunica intima on the vein. This dark purple is the tunica media, and then this looser structure around the exterior is the tunica externa. Due to having a much larger or much more muscular tunica media, the artery in a histological preparation, so we take this, we cross-sect it, and we slap it on a slide, it is able to maintain a circular shape with a circular lumen. A vein, when shown on a histological section, is going to, this one's actually wider than you normally see. Normally the lumen is very crescent shaped, kind of more even like this. And it just, the walls collapse in on that lumen. For arteries, move this a little. Okay. So I actually really like the way that this is diagrammed out, how it's shown. For each of, we're going to be talking about, there are 3 groups of arteries, and they're divided by size and function. So arteries have a very strong structure-function relationship. Their tissue composition really determines, is well suited for their function. So we'll start by talking about the elastic arteries. The elastic arteries are going to be the ones closer to the heart, like the aorta is the prime example. It is going to have the greatest proportion of elastic tissues of any of the arteries. It still has a substantial smooth muscle layer. And then this is the connective tissue on the outside, or the tunica externa outside. Okay, so elastic arteries are going to act as pressure reservoirs. So when the heart beats, that's kinetic energy. And so that's when the heart is in systole, it's beating. We're going to talk, because we're talking about the aorta, we're going to talk about the left ventricle. So left ventricle contracts. That's kinetic energy. But then it goes into diastole, goes into relaxation. Well, so that our blood flow is continuous and not on, off, on, off. The kinetic energy is stored in these elastic fibers of the elastic arteries. And so while the heart is resting, while it's in diastole, this elastic energy gets reconverted back into kinetic energy. So it's like stretching a rubber band and then letting it go back to its normal size. And that is going to assist in continuous flow of blood downstream. Okay, so while the heart is resting, while it's in diastole, the elastic fibers release their elastic energy and that's going to help propel blood in a continuous fashion. Elastin is found in all 3 tunics, mainly the tunica media. It does contain substantial smooth muscle, but it is inactive in vasoconstriction. So you're not going to get vasoconstriction at the level of the aorta or abdominal aorta. So that very large elastic artery, its function is going to be to, especially in the aorta and ascending, the aortic arch is going to be to convert kinetic to elastic and elastic back to kinetic for continuous blood flow. Okay. Then we're going to look at these muscular arteries. So the muscular arteries have much less elastic tissue, and per their name, they have a considerable amount of smooth muscle. They also have a fair amount of fibrous collagen fibers, and this will allow for them to anchor to structures, but also for other vessels to run alongside them. Okay, so the muscular arteries are going to be what we consider our named arteries. So we'll have a renal artery, a hepatic artery, iliac artery, a femoral artery, a brachial artery. So these named arteries, most of them are going to be in that muscular artery category. They are going to be distributing arteries. They deliver blood to body organs or like body structures, like femoral artery would be the femoral region. So regions or body organs. They do have the thickest tunica media with lots of smooth muscle, but they are active in vasoconstriction, but to a lesser extent of arterioles, which is where we're going. So if there is a large stimulus, like a large blood loss or damage to tissue, then they will participate in vasoconstriction. But vasoconstriction is really going to be controlled by the arterioles, the next level down. The muscular arteries, their main function is distributing blood to body organs and regions. Then we're going to finish off with arterioles. So arterioles are much, much smaller than a muscular artery. So these are not drawn to relative size. It says here size relationship are not proportional. These are, so this is a millimeter, this is a micrometer. There is 1,000-fold difference between these two units. Okay. So arterioles are much, much smaller. Of course, they have an endothelium, some elastic tissue that's mainly going to help hold things together, a little bit of muscle, and that's because they play the primary role in peripheral resistance, and then the tunica externa. So arterioles are the smallest of the arteries, and they vary a lot in size. You go from, there are large arterioles, a medium arteriole, and a small arteriole. The larger arterioles are going to come off of those muscular arteries, and they will contain all three tunics. Then as they reduce in size, because they'll branch off into other arterioles, then these are going to get down to a single layer of smooth muscle surrounding endothelial cells. Arterioles are also called resistance arteries or resistance vessels because they are very susceptible to vasoconstriction and vasodilation factors that control it. So arterioles are under control from the sympathetic autonomic system as well as local. So for local control, we have what are called autocrines and paracrines. They're hormones that feed back onto the cells that produce them. And these local controls will either, they can increase calcium levels, which cause vasoconstriction, or they can release things like adenosine that cause vasodilation. The specific local factors are not in your textbook, so I won't ask you like, oh, what is the effect of adenosine, or unless we get to it later on and it becomes a huge topic, which I don't see happening. So just know that you have both nervous stimulation and local chemical / hormonal control. So both of these factors act on arterioles to distribute, well, I won't call it distribute, but it is to distribute blood into certain capillary beds. So when capillaries are tiny, they're only 10 micrometers wide. When you go from something like a muscular artery that's distributing large amounts of blood to arterioles down to the capillaries, that is going to greatly decrease diameter and increase length. And both of those together greatly increase resistance. So the function of arterioles is to control the flow of blood into capillary beds. It is subject to both sympathetic autonomic nervous activity as well as local hormonal and chemical factors. So actually, we're going to talk about veins and we'll come back to capillaries. So veins are going to carry blood toward the heart. In general, they are deoxygenated in the systemic system. And on the other side of capillaries, capillaries feed into something called a post-capillary venule. So a venule is a very small, thin vein. And again, you can see this is measured in micrometers instead of millimeters. So these are around the same size as an arteriole, and you want that even distribution in size. So you go big to smaller to capillaries, and then smaller back to big. We'll talk about pericytes. Pericytes are smooth muscle-like cells that stabilize a venule. Larger venules have 1 or 2 layers of actual smooth muscle, and then veins have a thin tunica media, but the tunica externa is thick. Then we'll come back to why they're considered capacitance vessels in just a second. Okay. So as we go from arterioles, from large to medium to small, then they are going to merge into a capillary bed. Capillary bed is made up of capillaries. Their average diameter is 10 micrometers, which is this. So a red blood cell is at 7. So they tend to be slightly larger than a red blood cell. They are tunica intima only, so simple squamous epithelium or endothelium with a connective tissue basement membrane. But they are stabilized by something called pericytes, not parasites like the things that can, make you ill, but pericytes. And pericytes are spider-shaped, smooth muscle-like cells. So they function kind of like smooth muscle, but they don't form a layer. They stabilize capillary walls in more or less extent. They will help control permeability. They can get further apart to allow cells to be more permeable, or get closer together for cells to become less permeable. They can play a role in vessel repair. But the main thing is that they're going to stabilize these capillary walls, because a single layer of squamous tissue would be very fragile on its own. It's okay when we get to the pulmonary system because it's a lower pressure system. But on the systemic side, it's considered a high pressure system. And so we don't want to be busting through capillaries all the time. So these pericytes will help to stabilize those capillary walls. Capillaries reach almost every cell except for cartilage, epithelial, cornea, and the lens of the eye. This is why cartilage is so slow to heal, but it can get, it gets its nutrients and waste from fluid that is produced by the synovial membrane. So a little bit different. And the cornea gets it from the aqueous and vitreous, mainly the aqueous humor. I digress. Okay, so back to capillaries, stabilized by parasites. Its functions. The big one is it is going to be the site of gas exchange, nutrient exchange, and waste exchange between the blood and interstitial fluid. All capillary endothelial cells are joined by tight junctions. However, those junctions are what are considered incomplete. So it's not like the epithelium in your fingers to make it watertight. These are tight junctions, and then but they're not continuous. So in between, we have intercellular clefts. And this will allow the passage of fluid and small solutes. There are 3 types of capillaries. They are going to go from a little leaky to more leaky to really leaky. Okay. So continuous capillaries are by far the most abundant. These are the ones that are found in the skin, the muscle, the lungs. I don't know why it has CNS. We need to take that out. Okay, skin, muscle, lungs. They have incomplete tight junctions, and they have with small intercellular clefts. The exception is the brain has continuous tight junctions because that's part of the blood-brain barrier. But in general, they are going to have incomplete tight junctions to allow a little bit of fluid to come out. But they are abundant. They are found in the skin, muscle, and lungs. These are kind of the big ones. Okay, so this is just picture this section on the 3 type of capillaries. I don't think it's actually like in the text, it's in a figure that's either embedded in the text or off to the side. So it's a little harder to find if you're actually in your textbook. So continuous capillaries, least permeable, most common. You will find them in the skin and in the muscle. So good synopsis. Then we're moving on to fenestrated capillaries. These are found in areas of active filtration, absorption, or hormone secretion. I really like using the kidney as the example because the kidney has what's called an afferent arteriole, an efferent one, and then it has these capillaries, these fenestrated capillaries, and the pressure of this that goes through this capillary bed combined with having fenestrated capillaries allows filtrate to be produced. So we get this pressure, arterial pressure, going to these fenestrated capillaries, which have larger gaps, more gaps and larger gaps. And that's going to allow more plasma minus plasma proteins, that's why it's called filtrate, to be forced into this glomerular capsule. And then it's going to go into the kidney tubules to reabsorb some items and excrete others. So the kidneys, I think, are the best example because that glomerulus and the glomerular capsule, which we'll study under the urinary system, are just a great example. You'll also find fenestrated capillaries in the intestines and in endocrine cells that are around endocrine organs that are secreting hormones. So this is our middle one for increased permeability. The third one, okay, so sorry, recap, large fenestration, increased permeability, occur in special locations such as the kidney, the small intestine. Again, good sample, good summary. Okay. Sinusoidal capillaries are going to be leaky capillaries. They're very, I'm going to show you them. They've got, so these fenestrations are much larger and the clefts are much larger. So this is considered a sinusoidal capillary. It almost goes beyond being permeable. It allows access from cells that are outside to actually go into the bloodstream, like macrophages, and pull things out, like bacteria. It also will allow like B lymphocytes that to figure out if there's an antigen present. So these fenestrations are so large that they allow access in and out of the endothelium, which is a kind of a really weird, we don't really think of capillaries as being accessed from both sides physically. It's usually through diffusion gradients. The other thing though is that these fenestrations create a lot of turbulent flow because the blood instead is used to going along a nice smooth surface, and these are so large that they kind of catch things up, and they're going to result in what's called sluggish flow. And this sluggish flow allows those macrophages to access the bloodstream. It also, we're going to talk like in bone marrow, those megakaryocytes, so the ones that produce platelets, they put cytoplasmic extensions and they get kind of ripped off and they'll release and seed platelets. So it's a very unusual capillary. So it's not very common. It is by far the most permeable. You're going to find it in special locations in the liver. The liver is going to actually access this blood and modify it. The bone marrow, so that we get also reticulocytes get secreted. And then in the spleen for antigen presentation. So very specialized locations for these specialized purposes. So they are the most leaky and the most modified. They're the most unique. They're only going to be found in the liver, bone marrow, spleen, and I'm not real sure why also the adrenal medulla. These to me make perfect sense. This one not as much. Blood flow is sluggish, allows time for modification of large molecules. That'll be done in the liver. And blood cells that pass between blood and tissue is in the bone marrow and in the spleen. There are macrophages in the lining that actually access the blood and are going to capture and destroy whatever it finds. Okay, that's foreign. So a capillary bed is an interwoven network of capillaries. It creates what's called a microcirculation, which is the flow of blood from an arterial to a capillary to a venule. So this is a microcirculation. We have an arterial, so it starts with this terminal arteriole. This terminal arteriole is under sympathetic nervous activity control and local chemical and hormonal factors. It then is going to lead into this precapillary sphincter. And this precapillary sphincter is only under what's called local control. It doesn't have, it's not going to be under, there's no nervous innervation here. So precapillary sphincters are only under local control. And the precapillary sphincters will determine whether or not this capillary bed is open, so free flow to the postcapillary venules, or they can close this off completely where the whole thing would be white because there's no blood flowing through it, or they can, in some capillary beds, like in the mesentery, there is this thing called a thoroughfare channel where the blood flow will go from arterial to venule, but there's no diffusion of nutrients. It is outside of where nutrients are exchanged. So it just allows kind of this blood to flow through. So precapillary sphincter, kyphosmooth muscle, but it's controlled by local chemical conditions. It will regulate blood flow into the capillary bed. There are 3 options. True capillary, all the sphincters are open, great diffusion, great exchange of nutrients and waste, and that's how you would kind of expect it to be. Or it can be closed, and closed with no bypass or closed with a bypass. When things get closed, this is often when we are redirecting blood, like during exercise. These would open in the muscle, and then these would close in the small intestine, large intestine area, the mesentery. So, and then sometimes they just go through what's called a thoroughfare channel. So 3 ways. Once we go from capillaries, we go to venules and then to veins. Veins are going to return blood to the heart. So as I mentioned earlier, the veins have all the tunics, but they don't have a lot of smooth muscle. Okay, very little smooth muscle. So in order to keep blood flowing, blood flows because of a pressure gradient. So we need to go from the change in pressure. So we go from high pressure to low pressure. Blood always flows down the pressure gradient. There are some modifications to veins that assist with our high pressure to low pressure system. And one is to reduce resistance. So in a blood vessel, we have the walls and then we have the lumen. Along the walls are turbulent flow, which is slow. But in the middle, we have what's called laminar flow, and it is fast. Veins have a large lumen, and so they have a greater diameter for laminar flow. So by having this large lumen, they maximize the laminar flow and minimize this turbulent, slower flow. So they make it easier for blood to flow through a vein. The other thing is that veins are going to have valves, but not all veins. We'll talk about that in a second. So most veins have valves to ensure that blood flow is going to be unidirectional. They're called capacitance vessels because they'll hold 60 to 65% of the blood. This can be changed. We'll learn about that. I don't know if we'll do it today, maybe on Thursday. Okay, so the two modifications of veins to keep blood flowing are a large lumen, which is going to optimize laminar flow, and then having valves keeps blood flowing in one direction. Interestingly, the inferior vena cava, the superior vena cava do not have valves. Movement in both of those vessels is controlled by the alternating pressure caused by respiration in the abdominal and thoracic cavities. So it's kind of, I think that's kind of a fun and interesting fact, because you would think that the larger the vessel, that you definitely have valves, but in inferior vena cava and superior vena cava, you don't need them. You get the pressure differential through respiration. But most veins do have valves. So this is a diagram showing where at rest where blood volume tends to be. 60%, 65%, somewhere in there, it will be in systemic veins and venules, about 15% in arteries and arterioles. We have, this is the pulmonary blood supply, the heart, and the capillaries. This is a diagram of showing the heart being the pump, the vessels being the conduits, and we go from elastic arteries to muscular arteries to arterioles to capillaries and capillary beds to venules to veins to larger veins. On both the arterial side and the venous side, there are things called anastomoses. These are interconnections of blood vessels such that a region does not rely on one system only. Redundancy, or these collateral channels, are very important for maintaining blood flow and then for getting blood flow back to the heart. So arterial anastomoses are interconnected arteries feeding an organ or a region, allowing for collateral channels. These are common in joints where blood flow can get cut off due to movement, abdominal organs. And then the one that we studied in lab occurred in the brain. So in the brain, we have the circle of Willis. And off of the circle of Willis, you actually have a lot of other vessels that we didn't cover in lab. We covered the circle of Willis, the basilar artery, and then the vertebral arteries. But these arteries that come off the circle of Willis go to feed the lobes of the brain. So this circle of Willis is an example of an arterial anastomosis. Venous anastomoses are very, very abundant. Okay. They're so abundant that occluded veins really block blood flow. So when an individual gets varicose veins, those valves have stopped working and they become leaky. So that blood is now it's either sluggishly flowing or it's not flowing very well. Well, they used to remove them, but now they heat seal them. And so the walls collapse into this heat sealed vessel, which now there's no flow in or out of. And eventually the body will absorb that. But you can take out or heat seal pretty large vessels and it doesn't cause an issue in venous return because there are so many other pathways that the venous circulation can take. So arterial anastomoses. An example is circle of Willis in the brain, and this is going to provide for redundant blood supply. It also occurs in the heart. We didn't go over that when we went through the coronary circulation. Venous anastomoses are really, really abundant, and so an occluded vein is rarely going to affect venous return. Okay, so now we are getting on into kind of like the meat of the chapter, so the focus of the chapter. So the chapter is now moving from structures and the anatomy to more of the physiology. So what is blood flow, what affects blood flow, and how is it controlled? So blood flow is the volume of blood flowing through a vessel or organ or entire circulation in a given period. So blood flow is a rate, it's a volume per unit time. The blood flow that you have been introduced to is cardiac output. And cardiac output is considered the blood flow of circulation, so of the entire circulatory system. So volume per unit of time. Blood pressure is force per unit area. And it is expressed in millimeters of mercury. So if a common systolic blood pressure is 120 millimeters of mercury, and that is the weight of a column of mercury 120 millimeters high. It's kind of an archaic measurement, but it hangs around today. So that's what we use. The pressure gradient is the driving force to keep blood moving through the circulatory system. So blood flow and blood pressure. First, we're gonna. Okay. This relationship of flow is equal to the change in pressure over resistance. So blood flow is directly proportional to the blood pressure gradient. If the blood pressure gradient goes up, blood flow speeds up. Blood flow is inversely proportional to peripheral resistance. So if resistance increases, blood flow decreases. So now we're going to look at the factors that affect the change in pressure, and then factors that affect the change, that affect the amount of resistance. So resistance is sometimes called total peripheral resistance when we're talking about the circulatory system. It is the opposition to flow. It is created by friction of the vessel walls. And there are three sources of resistance that we talk about regarding the circulatory system. The first one is blood viscosity. And the greater the viscosity, the less easily molecules move. And so increased viscosity equals increased resistance. And I think that most people can understand how this would happen. If you have a glass of orange juice and you have a milkshake, you use the same straw, you can drink the orange juice much quicker because it will flow through the straw much more easily than that thick milkshake. So if you increase blood viscosity, you're going to increase resistance and decrease flow. Then the next two are total blood vessel length and blood vessel diameter. For total blood vessel length, the longer the vessel, the greater the resistance encountered. There's more area to cover and to have to overcome And so that's the greater resistance that it's encountering. It's basically encountering more arterial or what capillary wall. And then these two remain pretty consistent. So we tend to have a pretty consistent blood viscosity. Dehydration, it might change a little bit, but in general, our blood viscosity is pretty constant. Our total blood vessel length also remains pretty constant. And so that leaves this third factor, blood vessel diameter. This is the one that we can change. And so it is going to have the greatest influence on resistance. We can cause vasodilation, vessel gets bigger, resistance decreases, or vasoconstriction, diameter gets smaller, and resistance increases. And this is going to be due to that difference between turbulent flow and laminar flow. So if we have a vessel and it is this size, you get turbulent flow along the edge, which is slow. And then you get laminar flow in the lumen, which is faster. If you vasodilate, you make that larger, you still have the same amount of turbulent flow along the edge, but now you have a much greater area for laminar flow. Likewise, if you vasoconstrict, you are going to decrease the diameter and decrease the amount of laminar flow. Okay. So blood vessel diameter is going to be affected by that laminar flow versus turbulent flow ratio. Okay. Resistance varies inversely with resistance varies inversely with vessel radius. So radius increases, resistance decreases, radius decreases, resistance increases. In this example, the milkshake in 2 different straws, you're looking at 2 different factors. So the tall, thin straw has a longer length. It also is going to have a smaller diameter. And this is all going to create an increased resistance. The shorter, wider straw is going to have, so it's shorter vessel length, and then it also has a wider diameter, and both of these factors combine to decrease resistance. So if you're drinking a milkshake and you have a choice of straws, if you want to make it last, then you would take the longer, smaller diameter straw because your flow would be much slower. If you're in a hurry, you're hungry, thirsty, and choose the shorter, wider straw because it will have decreased resistance and you can access that milkshake much more quickly. Oh, we're doing good on time. Okay. So flow, and I wish they would use something other than F because F is usually force, but in this case, F is blood flow, is directly proportional to the change in pressure gradient over resistance. We've talked about the 3 factors that affect resistance and how changing blood vessel diameter is going to be play the greatest role in affecting resistance. So now we're going to look at the factors that affect the pressure gradient. So the heart is the pump. So when it contracts, it is going to impart kinetic energy to that fluid, and it's going to flow through the vessels. Pressure results when flow is opposed by resistance. So systolic pressure is the highest pressure that is produced in the arterial system. It's when the left ventricle contracts and it forces blood out through that aortic semilunar valve into the aorta. So that pressure is going to be the highest pressure produced. Then the heart goes into diastole, and at rest, that pressure drops, okay? Because the heart isn't pushing the blood out anymore, it's resting and relaxing. There is a typo here, okay? This should be pulse pressure. And pulse pressure is equal to systolic pressure minus diastolic pressure. So it's the difference between systolic and diastolic. So in this one, pulse pressure is 40. So 120 minus 80. So why do we care about pulse pressure? Well, if you look at this graph, we're going to first look at what is the amount of pressure, what is the force that's going to be used to keep blood flowing through the system. Is it 120 millimeters of mercury? Is it 80 millimeters of mercury? Is it 100? Do you just average the two? And it turns out none of those answers are correct. The amount of pressure placed into the system is going to be mean arterial pressure. So mean arterial pressure is the pressure that propels blood into the tissues. At first glance, you might think you just average systolic and diastolic, but that's close, but not quite right. The heart spends more time in diastole, so it's not a simple average. It's going to be calculated by adding the diastolic pressure plus 1/3 of the pulse pressure. So using that previous graph, the diastolic pressure is 80. The pulse pressure is systolic minus diastolic for 40. 1/3 of 40 is roughly 13, a little over, but that's okay. So to get mean arterial pressure, you add diastolic plus 1/3 pulse pressure. So the pressure that is propelling blood through the system is 93 millimeters of mercury. So it's slightly less than if you just average systolic and diastolic. You do need to know how to calculate mean arterial pressure. There will be a mean arterial pressure question or two on exam number 2. So you will need to know how to calculate that. Pulse pressure and mean arterial pressure both decline with increasing distance from the heart. So that is shown here. We get the largest negative slope in arterioles. So arterioles are high resistance vessels. So they are going to drop this pressure down because they're going to resist the pressure, resist that flow. So systolic, diastolic, pulse pressure, and you get the steepest decline in arterioles. So arterial blood pressure is determined by two factors: elasticity of the arteries closest to the heart. So these can store that kinetic energy and give it back, as well as the volume of blood being forced into them at any time. That would be the factors that affect stroke volume. Blood pressure is pulsatile. It rises and falls with each beat. Systolic pressure is that peak pressure. So it's the highest pressure. It's exerted in the aorta during ventricular contraction. It imparts kinetic energy that stretches the aorta. But so it is, systolic is peak pressure. The average is 120 millimeters of mercury. It's what is considered what you should be shooting for ideal function. I know some people are going to be lower and some people are going to be higher, but so that's the average. Diastolic is the lowest aortic pressure. It is after the aortic valve closes and the ventricles are in diastole, they are resting. Pulse pressure is the difference between systolic and diastolic, and the pulse is what we actually can physically feel. Okay. So if you're given a graph, like kind of the one before, it would have different values. Be able to identify systolic pressure, diastolic pressure, calculate pulse pressure. Also, know how to calculate mean arterial pressure. There's, I would say almost like for these few slides, there's maybe a disproportionate number of questions on these because there's a lot captured in these few slides. Capillary blood pressure is going to start around 35 and it'll be around 17 at the end. And so this is considered pretty low pressure, and that's good because high blood pressure would rupture these capillaries. And also the capillaries are permeable. So if you increase the pressure, you're going to drive a lot more of that fluid out, and we would get edema and a decrease in venous return. So good low blood pressure in the capillaries is good. Venous blood pressure. Venous blood pressure is pretty low. It's only around 15 to 20, depending where in the system you are. So venous pressure is too low to permit adequate venous return. We do have those adaptations of having a large lumen, so it decreases resistance, as well as the valves to ensure one-way flow. But those two adaptations are not sufficient for adequate venous return. So we have 3 additional adaptations, the muscular pump, the respiratory pump, and then sympathetic venoconstriction. The muscular pump is the contraction of skeletal muscles against the veins, will help to provide the pressure for them to go up to the next valve and up to the next valve and up to the next valve. So skeletal muscle contraction is going to help in venous return. And then in exercise, it creates a lot, it greatly enhances venous return. So that's shown here. When it contracts, it helps to propel blood up to this next valve. The respiratory pump As I mentioned, this is really important for like the superior vena cava, the inferior vena cava. Pressure changes during breathing. It's an alternating with abdominal and respiratory pressure. We'll get more into that with the respiratory system. But for now, you just need to know that it's that alternating in cavity pressure that helps to pump blood through the inferior vena cava and superior vena cava. Sympathetic vasoconstriction, sympathetic control, smooth muscles constrict and assist blood moving back to the heart. We are going to stop here because this is quite a large section and I don't want to start it yet if I can't finish it, which I can't. So we're going to stop here. So don't forget that you have that exam 1 is open till 10 on Saturday. I will say I couldn't get it to accurately, when it auto grades, it's not completely accurate. So if you get a score, let's say you get a 70, when it auto grades, it will probably, I need to go back and hand grade a few things. And it will then, so you might see it increase. Okay, so if it comes at, you get a 70 on the auto grade, don't be surprised if it changes once or twice as I adjust things, but it'll only go up, it won't go down. So whatever score you get, it will, and it was not going to go up over probably 5 points. Okay, there's just, so, but if you see changes in a few points, that's me. Okay, so I know it's a little aside and a little maybe confusing, but it works in your favor. Okay, so are there any questions on the material or on previous material or anything that you guys want to go over?
Michelle, you said that nothing that we just went over is going to be on this exam.
Correct. This is all on exam 2.
Okay, thank you.
Anything else? Okay. Well, then I will see you on Thursday.